All right, it's going to be a hand-waving talk. So, so I want to bring you to the beginning of science, and start from there, from the very beginning. And the first person who called himself a scientist, philosopher, was Pythagoras. And Pythagoras lived 2,500 years ago. This portrait was made 300 years after his death. We don't know how he looked like, but we know what he said. So he said all things are numbers. That's one thing he said. And second thing he said, there's a harmony of the universe, the musica universalis. So he also came up with the Pythagoras theorem and the number pi that can be derived from that theorem. Pi is, of course, the first letter of Pythagoras. And for a long time, people believed that pi is an irrational number, so the decimal expansion of pi is random. There are a number of papers published how random is decimal expansion. Until someone, our colleague, Professor Puent, has found mapping. So he found a mapping that actually showed an order in that chaos. That's published in a journal called Complexity, 2001, 25 years ago. So if 1 out of 10,000 bits is randomized, the whole picture disappears. So it's a order that is spread all over, and you need to extract it by proper mapping. And now we go back to Stockholm, to Karolinska Institute, where I work. By the way, this clock hasn't started yet. No. So I still have 45 minutes to tell you. The greatest scientist there was Jon Berzelius. Here he is. And he was a sort of mass spectrometrist because he created, and that was 200 years ago, he created a table of elements with the masses. Of course, these were average isotopic masses. These were chemical masses. And then Dmitry Mendeleev, some 40 years after, looked at this table and saw order in that chaos. He took this single-dimensional table of Berzelius and created a two-dimensional table, periodic table. He saw periodicity in masses, okay? Now we know that these are actually atoms, electronic structure, and we know also that they are isotopes. Actually, it's not elements, these are isotopes, and there are lots of them. So let's look at these isotopes, which are the most important isotopes for life: hydrogen, carbon, nitrogen, and oxygen. Here you have masses, abundances. Where is the water? Can you see it? Not easily, right? So we need proper mapping. And proper mapping can be derived. So we need 2 dimensions to present it in 2-dimensional view. We need 2 independent dimensions. And we came up by, they call it serendipity. Serendipitously, we came up with this mapping. When the first dimension is a normalized mass defect, is the difference between the monoisotopic mass and the average isotopic. I don't have a pointer here. Well, anyway, so I cannot point, sorry. The second dimension is the difference between average mass and monoisotopic mass. And these are normalized by the nominal mass, and nominal mass is integer. So when you do that, you have these 2 dimensions, and the first dimension relates to these decimals after the integer point in the mass of the most abundant isotope. And the second one refers to the abundance of that isotope. So these are 2 independent things. However, when we put 300 molecules, these are peptides, on this map, we see an order all of a sudden. Instead of galaxy, which is a random distribution as we expected, we saw this line. And first we thought it was an error, it was a bug, but it was actually a feature. And we looked at this feature and found that the molecules on the line, they follow this equation, Z equal to 0. And If Z is below 0, then the molecules are below the line, and above the line is positive. So what is Z equal to 0, this, for which you have this straight line? We call it a resonance, isotopic resonance. It turns out these are the molecules which have the simplest possible structure. So if you look at all molecules with carbon, hydrogen, nitrogen in meteorite and plot them by their Z value, you see the Z equal to 0 is at the peak of the distribution. It's a critical point of the distribution. This is the simplest molecules, the most abundant molecules in the universe. Okay, and for them, we have this resonance, and it took us time to understand what it all could mean, and we came up with isotopic resonance hypothesis, basically based on the assumption that less complex molecules are working faster. And isotopic resonance reduces the complexity of molecular systems composed of hydrogen, nitrogen, carbon, and oxygen. And the complexity reduction leads to faster kinetics. And early life on this planet depended upon that, so it emerged much earlier because we have this isotopic resonance. That was a hypothesis. You need to test it. And we tested it by looking at the literature and making some non-trivial predictions. Some of the non-trivial predictions were that if you have two systems, which are, one of them is growing faster than the other, but otherwise they are in the same conditions, then the faster growing system should be closer to the resonance. And we predicted for each element how much the isotopic shift will be, and then found a paper where the shift was measured between fast-growing pigs and slow-growing pigs in the same farm. And the correlation between the prediction and the measurement was embarrassingly good. So we realized that we're doing something right. It took us some time to publish a paper where we did experimental verification of this hypothesis. And we made a number of predictions and then verified them. And one of the predictions, was that at 3.5% nitrogen-15, which is about 10 times more than normally, you would have a faster growth of the cells. And that's what we saw. And when I presented this result, one of my colleagues, Helena Gorochova from Stockholm University, she said she didn't believe it. And she actually spent some time to verifying this. And lo and behold, she also found these resonances and published a couple of papers on that subject. I need to find a proper point in the universe where this thing works.
You had it.
Had it. Okay. Yeah, so now deuterium. We go back to deuterium, and of course, deuterium is due to Harald Urey. And when we thought, what can we predict about deuterium? Now, we predicted that if you increase the concentration of deuterium about twofold, then this line becomes infinitely fine, and so the growth rate should increase. And the opposite is true. If you decrease the amount of deuterium compared to normal, 150 ppm, then the growth rate will slow down. And we thought, people must have found this long ago, so we started to look in literature. And the first literature source I found was Valentin Lobyshev's. Valentin is here?
No, unfortunately not.
Unfortunately not. He was in the, unfortunately. So he was head of the biophysics department in Moscow State University. In the 1970s, they done a lot of studies on deuterium, and they found this peak at 400 ppm, which they could not explain. This is simple reaction, sodium potassium ATPase reaction. And of course, no theory explains that except for the isotopic resonance theory, but it wasn't known then. And then, of course, people pointed out to Gabor's work and his seminal paper when they increase the amount of deuterium and the growth rate of the cells increased, and they decrease it, and the growth rate decreased, and that was the basis of deuterium-depleted water. So some time ago, we decided to test the molecular mechanism of deuterium-depleted water. So we found cells, A549 cells, which, by the way, are currently the most frequently used cells in biology. They're more used than HeLa cells. So, and it turns out that they're very sensitive to deuterium content, and at about 80 ppm, the growth rate decreases by 30%. That was the strongest effect we saw. And we came up with a molecular mechanism which is based also based on mitochondria and reactive oxygen species, ROS. And it was also held to publish, but okay. And then we went in the opposite direction. We increased the amount of deuterium, and of course we expected the cells to grow faster, as Gabor has shown and people before us. Actually, that things grow faster when you add more deuterium is known since 1930s. And then we realized that deuterium is actually a cell growth regulator in the region between about 30 to 40 ppm to about 350 ppm. Okay, but what if you step outside that region? For instance, you go to Mars. On Mars, there are about 6-7 times more deuterium in the environment than on Earth. And that already on declining curve, so it should be inhibition. And people talking about sending people on Mars, like Elon Musk. Isn't he worried about deuterium there? By the way, on Mars there is no isotopic resonance, and therefore, according to our hypothesis, life on Mars is unlikely. So we did this experiment. It took us 2 years. We created a system. We already bought this system. It existed. There's a small organism, 2 shrimps, grown in isolated environment, and for 2 years they grew in the environment where everything was the same except for water, or deuterium containing water. And what we found that at about 600 ppm, the survival of this shrimp is actually declining compared to normal composition. And that should make Elon Musk very worried. And I told my group, half joking, that I'm waiting for Elon calling me any day now and saying, Roman, what are you doing? You're shutting down our most important project. But he never called, actually. Then I realized the hard truth. Elon Musk doesn't read our papers. What is he doing? I don't know. Does he have anything more important to do? Very recently, actually this month, Stepan Jimak from Krasnodar, Russia, had published this paper, very nice. They refer to some of our work, but not to the study that I showed you before. And they also tested the environment with Martian composition of deuterium, and they also found some detrimental effects on mice. And then, of course, Laszlo Boros, who is here, and he came up with a datanomics idea, that is a branch of science to study how deuterium is distributed in nature and how it behaves and how it is playing a role in how we can use this knowledge in order to improve human health. And that's great. So I was analyzing why people are so resistant to the idea that deuterium manipulation in these ranges would change the behavior of the biological system. What is the culprit of that? Why there are 50 people in the room and not 500? So I came up with the following answer. It comes from the conventional wisdom, the wisdom that is spread in the scientific community, and it goes like that. Okay, if you increase the concentration of stable isotopes, heavy isotopes, to 100%, then maybe something will happen. But at normal isotopic compositions, that is below 3%, the stable isotopes are inert. They don't do anything, and therefore stable isotopic composition just reflect the biological processes, but not affect them. And of course, the possible exception is deuterium, but let's just address this problem by ignoring it. And that's the conventional truth that is the main obstacle as far as I can tell. And it is based on thousands and thousands of research papers produced by hundreds of research groups analyzing stable isotopes for the purpose of environment and ecology. And they want to link the isotopic composition of the food to the isotopic composition of the tissues. And originally, before 1970s, the received the wisdom was that you are what you eat. That is, the isotopic composition of these two should be the same. But look at plants. Plants eat CO2. This is isotopic composition of CO2, of carbon, and you see the plants have very different isotopic composition depending upon the type of photosynthesis, but within this type there's also spread. So you are not what you eat, and we also found that deuterium and proline and hydroxyproline of seals and collagen actually are twice as much deuterium as in seawater, and there is no way you can explain it by food. So you are not what you eat. And from 1970s, people modified this paradigm. It was due to the seminal work of De Niro and Epstein. And De Niro is not De Niro we know from the movies, and Epstein is not Epstein we know from the news. Well, anyway, they're two great guys from Caltech, but I think they misled the public greatly. What they said is that you are what you eat plus delta. So they introduced this delta, which is a constant. It's called diet tissue discrimination factor, and it may be different for different organisms, different tissues, but otherwise it is stable, doesn't change with the food. But if this is true, look at this equation. The slope of the linear regression between delta tissue and delta food should be exactly 1. So, and the contradiction to that paradigm already exists in their own paper. So they show this data set for mice, and you see there are 3 diets, and for 2 diets, the tissue shifted to the left in terms of carbon composition, carbon-13, and in 1 diet to the right. Right? So depending upon whether or not the diet had more or less C13 than -20 per mil. Remember this number, it will come back later. So how did these 2 great authors, explain this? They don't make any comments to that, so they leave it to the reader to discover. So this idea that by measuring isotopic composition, especially N15, you can determine the trophic level of animals is widely spread. And I said there are literally thousands of papers with hundreds of review papers based on this paradigm, with 3 to 5 per mil is separating one trophic level from the other. But if you look at the few studies where this is tested, you find a lot of inconsistencies like that. Here, look at the left one. There are 5 different diets, and it is expected, these were fish growing on these diets for like 40 days, so the turnover should be 100%, nearly 100%, and they expected the isotopic composition of the tissues, especially muscle and liver, metabolically active, to be very close to these 5 diets, and this is on the scale delta carbon versus delta nitrogen. But when they did the experiments, this is a middle slide, you see all isotopic compositions were focused into 1 point. And the deltas are vastly different. How do they explain that? What did they say? That the paradigm is completely break down? No. I mean, they invented a very inventive way of explaining the way but keeping the paradigm. So another data set, here is the stomach content of animals, marine animals, versus their tissues. And again, there is no correlation. There should be a straight line between these 2 dimensions with a slope equal to 1. Do you see a slope equal to 1? No, of course not. So what happens is the slope between delta diet and delta in tissue is not 1. What happens is that, as shown here, there's experimental data on the blood plasma in a control experiment with captured bears. There is a point where this line, and here it has a slope 0.4 instead of 1. It crosses the line with a slope equal to 1, and to the left delta is negative, to the right delta is positive. And when at the cross line, here at cross point, the delta is 0. So the point is that here in this control experiment, instead of slope equal to 1, the slope is 0.42 for carbon and 0.88 for nitrogen. It's not 1. So once again, to show that if you don't have a slope equal to 1, you always have a crossover, a crossing point, the equilibrium point where delta becomes 0. To the left or to the right, the delta changes sign. That means delta cannot be constant, period. And the error between the straight line with slope 1 and your experimental line will easily be 3, 5, 10 per mil, and that means your trophic level are all screwed up. So how is it in reality? We looked at 5 different studies, and we found them all showing that this equilibrium point is close to minus 20 per mil in the carbon scale, on the carbon scale. And this is the same like in the first paper by De Niro and Epstein. And then we looked at previous data. I mean, again, people should have seen it. And yes, they saw something like that. The great review paper by Kaut et al. And they found, they look at the different dimensions, but also saw an equilibrium. If I interpret their data, they also saw an equilibrium at minus 20 per mil. But people have criticized this paper and largely ignored that because they found one specific error, and therefore it was justified. This error was justified, but it was just a minor thing. But they because of that, they just said the whole thing is wrong. So what we did, we looked at literature using AI and all search engines. We found dozens of high-quality studies, and high quality means that they use at least 3 isotopically different diets, and the R-square of the line between diet and tissue is at least 0.8. So that means R is, the correlation is at least 0.9. And so you see here to the left 32 such studies, and they're showing a different position of equilibrium, but the average position is minus 20 per mil. And for nitrogen-15, the average position is plus 15 per mil. What is the message? And the message is, the only explanation of this effect is that the body resists changes of isotopic composition. And why? Because there is the isotopes play a role. They actually modulate their own fractionation. Okay, let's take the theory to extreme. And to the extreme means that remove all heavy isotopes. Only light isotopes are there. The isotope shift is 0, and all this galaxy becomes just straight line at 0. What happened then? And what happened then is something extraordinary, and that we know from the work of a Nobel Prize winner published 25 years ago. When they look at the formation of ozone, ozone has 3 isotopes, 16, 17, 18, or in their lingo 6, 7, 8. And there are 10 isotopomers, and 3 of them are fully symmetric monoisotopic, 16, 16, 16, or 6, 6, 6, 17, 17, 17, 7, 7, 7, or 8, 8, 8. And the formation, rate of formation of these 3 monoisotopic molecules are very different from the rates of formation of polyisotopic molecules, especially the one that is totally composed of totally different isotopes, 6, 7, 8. So therefore, the hypothesis is if we start to grow organism in purely monoisotopic media, then we will see some very significant and very strange effects. We should expect fast growing rate. And that's exactly what we saw. Supe Zhang in our group, she's done a lot of these experiments. So E. coli is easy to grow on minimal media, and minimal media is just one source of carbon and then one nitrogen, salt, and then just microelement and water. So you can manipulate the isotopic composition of nitrogen by the salt, carbon by the source of carbon, hydrogen, oxygen by water. And then we'll saw that when we deplete heavy isotopes, at least tenfold each one, the growth rate increases significantly. And then we do mass spectrometry. That's what we're doing for bread and butter. And we see the isotopic composition of proteins, which normally is broad, becoming squeezed one, and the monoisotopic peak is very tall. So that tells us that we have depleted heavy isotopes. Okay, and then we started to express in these E. coli different enzymes. you know, recombinant technology, and then the same bacteria can be grown in normal media, and we can extract these enzymes as well. So we can compare easily the monoisotopic enzyme, we call it ultralight, and normal enzyme. And we saw that ultralight enzymes work much faster. And how faster? We call it acceleration factor. And depending upon the temperature, the effect is increasing with lower temperature. But we can easily get a factor of 3 or higher. So factor of 3 higher rate, while the only thing you do, you remove 1% of all atoms. And of course, people wouldn't believe that. We had to explain this, and we explained that in great detail. We say that what happens when you remove heavy isotopes is that the molecule becomes pure and its conformational space becomes much smaller. That is, conformational entropy is greatly reduced. And on average, the form and the structure of the molecule will be very close to the optimal one, because the presence of every heavy isotope, it changes the structure a little bit, but most often than not, it changes it in detrimental way. So it's like mutations. Most mutations are deleterious. Only very few mutations are advantageous here as well. So if we remove all these heavy isotopes, like removing all mutations, and you have a pure breed of the molecules. So it was, again, not easily published, but fortunately, Angewandte Chemie was very perceptive, and after review, we did it in 4 different enzymes, and all 4 have shown an effect which also decreases with temperature, but at low temperature, there is significant difference between normal and ultralight. So this could immediately be used in technology. And then we went further, because E. coli are bacteria, and they're single cell. We wanted to do something with organism, multicellular, and the smallest organism that you can work with in laboratory is C. elegans. It's nematode, it's a worm, and it can grow on E. coli bacteria as the only source of food. So this is what we did. We grew 3 types of E. coli. One is normal, another is isotopically normal. One is ultralight, and the third one is heavy. And heavy means we added a little bit of deuterium, a little bit of C13, a little bit of nitrogen-15, a little bit of oxygen-18. But the amount of heavy isotope in any element is less than 2% still. So still it is within the normal range, so to say. With people would say this is not heavily enriched, this is lightly. This is similar to normal isotopic composition. And then we measured different parameters of the worm. The one thing that we were interested in is the length, and it turns out that ultralight are significantly larger. By significantly, I mean statistically significant, larger, and larger like by 5 to 10%. And they also move faster. When they're young, they move significantly faster. And the lifespan is much shorter. So they live fast, live short. Hippies would like that. Right, so we wanted to publish that, but then I said, look, this is a negative message. This is a negative message. You know, you can shorten life of C. elegans by many other ways, by toxins, for instance. So are light isotopes toxic? No, but we need to try to rescue that. So we try to rescue by taking ultralight embryos and putting them on normal food, and now yes, they live longer. And then there are counterparts which live on ultralight food, but not as long as normal, totally normal embryos living on normal food. However, the heavier ones that are born from heavy food parents and live on heavy food, they are living a little bit longer life than normal. So, there was a debate in literature whether heavy eaters live longer. By heavy eaters, mean isotopically heavy. This is a paper from Shepinov. He was an Oxford researcher. which is called "Do Heavy Eaters Live Longer?" And it created a lot of debate in literature. But yes, so basically that's what we found. We also did proteomics analysis in 2 dimensions. One was expression level analysis, another one was the solubility of proteins analysis. And then when we did principal component analysis, the first principal component separated the young ones from the older ones. So this component could be used as a pseudo-time. And we look at the data for normal and ultralight worms. We saw that if I interpret that in terms of time, the ultralight worms are born younger than the normal worms, and they quickly become older than them. So when they die, they're much older, so to say. And this is on both scales, which are independent of each other. That's pretty cool. So why is that? Well, this is because the lifespan of C. elegans is genetically determined. And if you increase metabolism, then you increase the rate of the internal clock. They they grow faster, they move faster, and they age faster. The question is whether this same applies to humans, and hopefully not. Hopefully not. Hopefully our aging is not genetically determined by the accumulation of debris or any other factor. Therefore, isotopes, we don't need to eat heavy isotopes. Complementing studies. People have looked at the role of stable isotopes well before us. For instance, Buchachenko, he came up with an idea of magnetic isotope effect. And there, in his view, the isotopes which have odd mass, they are fermions, not bosons, and they have magnetic moment, and they have deviating chemical properties which can be used in biology. And then there is another study, quite recent. There is a company called Metallomics, created by Peter Novak, Max Temnik, and the team, and they actually like zinc 64. Zinc has a number of isotopes, but the lightest one is 64. According to the previous research, Buchachenko, so this zinc 64 should not have any specific properties, but according to these guys, it has, and they try to develop it, and specifically a diaspartate of zinc 64. They formulate it as a drug, KLS1, and is now in clinical trial against a number of diseases, particularly against cancer. And they show that it works on mice fantastically. A bit of a problem is that the normal zinc contains 50%, about 50% of zinc 64. So the isotopic effect should not be that high. But, you know, nature sometimes works in an unexpected way, and we are looking forward to the result of these studies. And they already published a number of papers. We're looking forward to see more. And then the most forward-looking part is the isotope economy. What is the isotope economy? Some Dr. Johann Tenenbaum, he was a scientific advisor of somebody called Lyndon LaRouche. Maybe some in this audience remember this name. That's a bit controversial, but he was really a physicist, and he published this review, the isotope economy. Let's not go into the details of this review. That's mostly about radioactive isotopes, but the whole idea is this. Production of isotopes, that is separation of isotope, is the ultimate case of high technology. Why is that? Because the starting material costs next to nothing. You know, think about deuterium depleted water. What is the starting material? Normal water. How much does it cost, right? Next to nothing. But the final product, the separated isotopes, are very, very valuable. And so they're pure materials, they're ultra-pure materials, and ultra-pure materials have different property than pure materials. But there's a higher level of purity, which is isotopical purity. So isotopically pure materials have yet another layer of properties which are different from normal ultra-pure materials, and it needs to be explored. And isotopically engineered molecules, we can put light or heavy isotopes specifically here and there, and Mikhail Shepinov, I already mentioned him, the author of this paradigm that heavy eaters live longer. He has started a company when they produce fatty acids with the deuterium atoms put in specific positions, and that prevent them from oxidative damage, and they believe that this can be great for a lot of diseases. The jury is still out there. Clinical trials were inconclusive, but they are still hopeful. And then, so we have medicine, biology, agriculture, technology that is waiting for us to understand better how isotopes work and provide the recipes for new materials or improved processes, just like ultralight enzymes, which work faster. And we also probably need to control isotopic environment. We cannot just send people to Mars without thinking about what they're going to eat there in terms of isotopes, in terms of deuterium and other isotopes. We can also try to explore space and other planets by looking at their isotopic composition and see how well they're suited for human forms of life. For instance, on Venus, there's 100 times more deuterium than on Earth. It's 1.8%. That's deadly. That's deadly for us. You know, besides the 400 degrees Celsius temperature and sulfuric acid rains, you know, the isotopes will kill us if we survive that. And then astrobiology, today telescopes, radio telescope can actually look at the light of the stars passing from atmosphere of the planet and see signatures of small molecules that can actually measure isotopic compositions for carbon and nitrogen and deuterium and hydrogen without visiting these planets. And so we can calculate, based on this data, whether or not there is an isotopic resonance there. And if yes, then the planet is likely suitable for life. If not, sorry guys, don't go there. Choose something else. So what is the problem for all that vision? Why do we don't have this grand vision already now? So because we don't have this technology for separating isotopes here in Europe. Europe is so much backwards compared to the rest of the world, especially Russia, now also China, in separating isotopes. Who is separating isotopes in Europe? France, the only country, and occasionally from time to time, they don't have a sustainable production of any stable isotopes. So and therefore all the products on the market are imported to Europe from abroad. And because of that, European scientists are reluctant of studying isotopes. And because of that, there is no breakthrough and there is no efforts really in that. So let's hope that this will change in time. And I came to the conclusions. The first one is stable isotopes play an important role in biochemical reactions, modulating their rates. The effect of isotopic content is non-linear. There are resonance compositions at which reactions accelerate or decelerate beyond the expected trend. And deuterium is a cell growth regulator in the concentration range of 30 to 350 parts per million. Deuterium-depleted water slows down fast-growing cells, for instance, cancer cells. Probably has other properties. We'll hear more about that, therefore I don't focus on that. Excess of deuterium about 300 ppm, such as on Mars, may be harmful to terrestrial organisms, and future missions to Mars might take this into account. Even at natural abundances, heavy isotopes, not only deuterium, modulate biochemical processes, including their own fractionation. The old paradigms, that is, you are what you eat plus delta, which is constant, is obviously wrong. There appears to be an equilibrium isotope compositions at which there is no isotope fractionation between food and tissue. Removal of heavy stable isotopes, carbon, hydrogen, nitrogen, and oxygen, accelerates growth of organisms in metabolic processes, but may shorten the lifespan if it is genetically determined. Isotope economy is the ultimate case of high technology. Starting material costs is negligible compared to the value of the final product. Isotope economy is the future. With that, I would like to thank my group members, the former ones, the current ones, the funding agencies, and thank you very much for your attention.